In this activity, we're going to look at the negative stain, which is activity 3-6.
The negative stain, the purpose of it is to visualize bacterial cells and determine morphology, arrangement, and accurate cell size. So when we talk about morphology, morphology refers to shape. So what is the shape of the bacteria itself? Arrangement has to do with how the colonies are arranged, meaning when the bacteria grows, is it going to be an isolated cell? Is it going to grow as pairs? Is it going to grow in a chain? Is it going to grow as a cluster? And we'll talk about different types of arrangements in a minute. Now, you guys did your gram stain, and one of the things that we did in our gram stain was that we did remember a heat fix step, meaning that you guys put your bacteria on your slide, you allowed that slide to air dry, and then once it was dry, you did a heat fix by passing the slide through the flame 3 times. And remember that the heat fix step in the gram stain or in any other simple stain, the purpose of that is that the heat fix is going to cause proteins to denature, meaning it causes them to unfold, and it also causes those proteins to coagulate, meaning stick together. As a result of this, the bacteria will then adhere to the slide, and it will cause the bacteria to be killed. Why don't we use a heat fix step in a negative stain? And that's because the heat can cause fragile cells to shrink. And so, for example, if we wanted to determine accurate cell size, if we heat them, those cells are going to shrink, and it won't let us have an accurate measure of cell size. Additionally, fragile cell types like spirochetes or spirals are very sensitive and fragile. And if you do a heat fix step, it can cause those fragile cells to become damaged. And it would no longer allow you to visually see the appropriate type of morphology. And so the reason that we do a negative stain is that we don't use heat, and that's used to determine accurate cell size as well as proper morphology and arrangement. And so I have a question for you, and that is, why in this example is the background stained in a negative stain? So if you look at this picture here of a negative stain, notice that the background is stained, and you see these middle clear parts. Well, these clear parts are the cells themselves. So remember that if you're looking at a simple stain, right, a simple stain like a gram stain is also referred to as a basic stain. And in a basic stain, remember, what charge would you find on a basic stain? And the answer would be that you would find a positive charge on the stain. Remember when we talked about cells themselves, and we said that cells have what type of charge? And the answer is, remember that cells have a negative charge. And so in a simple stain, the simple stain has a positive charge, and the positive charges are attracted to the negatively charged cell. And so when we do a simple stain, we're going to be staining the cell and not the background. So now thinking about that, what do you think the negative stain has that causes it to stain the background and not the cell? And so first things first, let's start with if the stain is acidic or basic. So if we just said that a basic stain carries a positive charge, what type of stain do you think a negative stain would be? And the answer would be that it is an acidic stain, meaning that it donates hydrogen ions into the solution, and as a result, the stain is negatively charged, right? Because if it donates H+ to the solution, that's going to mean that the stain is going to carry a negative charge now. Now, if our stain is negatively charged, how will that stain interact with the cell? Do like charges repel or do they attract? And the answer is that like charges repel one another. So stain is repelled by the cell. Because again, the like charges are going to repel one another. They're not going to want to interact, and instead the background is stained and not the cell. And so some examples of a negative stain that we're going to use would be like Congo red. So Congo red we're going to use both in our negative stain and also in our capsule stain, which we'll talk about in a minute. We also have a type of negative stain called nigrosin, which is going to be black. There's one called India ink, eosin, etc. And what these stains all have in common, again, is that they are an acidic stain. They carry a negative charge on their oxychrome, and as a result, they stain the background and not the cell. So when we look at the bacteria, we're going to look at various types of morphology. And so some bacteria are going to appear coccus-shaped or plural cocci. Some bacteria are going to be rod-shaped in what we call bacillus-shaped. Plural would be bacilli. Some bacteria would be what's called spirillum, and these are going to be rigid spirals. Some bacteria are going to be spirals, but they're going to be flexible spirals. And this shape is called spirochete. Some bacteria are what we call vibrio, and vibrio basically means that they're curved rods. And so these are the basic common types of morphologies that you would typically see. If we look, so this up here is morphology. So again, the shape of the bacteria. Down here, we have the arrangement. And that is, how do the bacteria grow together or separate? So some bacteria are strepto. Strepto means chains. So you could get streptococcus, for example. So looking here, this is streptococcus. They're chains of coccus-shaped bacteria. So that's streptococcus. You could have streptobacillus, meaning that they're rods growing in a chain. Sometimes you'll see bacteria being staphylo, and staphylo refers to the fact that these grow as grape-like clusters. Whoops. No G again. Grape-like clusters. And so instead of being in chains, they grow as these little clusters. So for example, you could have Staphylococcus, meaning that you have lots of these grape-like lots of these round bacteria growing in clusters. The one you don't typically see, you're not going to see, is going to be Staphylobacillus. So no Staphylobacillus. And that has to do with the way that the bacteria divide. In bacillus, they're going to divide along the short end, meaning that they're going to form chains. They don't divide vertically. And so you're not going to find Staphylobacillus. You can find Streptobacillus, but not Staphylobacillus. Some bacteria are diplo. Di means two. So what we're looking at here is Diplococcus, two round-shaped bacteria growing together. You could see Diplobacillus, for example. And so you want to start becoming familiar with both morphology and arrangement. So What's an example of a medically important bacteria that can be seen with a negative stain? And so typically remember that the reason we do a negative stain is to visualize cells that could be fragile due to a heat fix step. And a type of bacteria that you'll typically see with this would be your spirochetes. And spirochetes, like what you're seeing here, spirochetes are going to be spirals, but again, they are flexible spirals. They're not a perfectly tight spiral. Notice you can see it almost has kind of a wavy appearance. That's a spirochete. And if you do a spirochete using a simple stain, the heat fix step might affect the shape or the morphology. And so instead, it's best to visualize a spirochete using a negative stain because the negative stain lacks that heat fix step. So an example of an example of a bacteria seen with a negative stain would be Treponema Pallidum. Treponema Pallidum is the organism that causes the sexually transmitted infection, or STI, that causes syphilis. And so how do you get syphilis? Again, it's a sexually transmitted disease. So through some sort of sexual contact. When we look at these organisms, one of the first signs of syphilis is known as what's called a canker. And so what you're looking down here is you're looking at this canker, this clean, kind of painless, ulcer-like appearance on genitalia. So an example in males, this could be found on the penis. It could be found on multiple sites. It could be on the vagina. It could be orally. But basically, one of the first signs of syphilis is going to be this canker. And this forms at the site of entry where the organism enters the body through the mucosal membranes or breaks in the epithelium. If you see multiple cankers, that means that typically more than one organism entered the body. And so the canker basically starts out as a local infection by Treponema Pallidum. And so this canker could be accompanied by swollen glands. They may last from 1 to 5 weeks, and they may even disappear by itself if no treatment is received. Now, that doesn't necessarily mean that that's a good thing. If you see these cankers and they disappear on their own, it does not mean that you're cured of syphilis. It could be that now your syphilis is going on to a later stage. About 6 weeks after the first sores appears, the second stage of the disease may start to appear. And the most common symptom during the stage is going to be rash, which can appear on any part of the body. Other symptoms during secondary stage of syphilis can be tiredness, fever, sore throat, headaches, hoarseness, loss of appetite, hair loss, swollen glands, etc. And these signs and symptoms will last typically between 2 to 6 weeks, and again, generally can disappear in the absence of adequate treatments. The third stage, which is what we call late stage syphilis, this is typically syphilis over a longer period of time, several years. Late stage syphilis can involve tissues like the skin, they can involve bones, the central nervous system, and heart. Untreated syphilis can lead to destruction of soft tissue and bone, can lead to heart failure, blindness, and a variety of other conditions which may be mild to incapacitating. And so people sometimes will have problems with the brain as a result of late stage syphilis. Once syphilis progresses to late stage syphilis, it's much more difficult to treat. It's best if syphilis gets caught at the early stage, typically at the canker stage of the disease, so that it can be properly treated, because this type of bacteria or this type of organism is a bacteria. Bacteria can be treated with antibiotics, and typically penicillin can be used to treat syphilis. And so if it's properly caught and treated early on, you can become cured of syphilis. Once it progresses to late stage syphilis, though, it's much more difficult to treat. Women with untreated syphilis can transmit the disease to unborn children. which can lead to death or deformity of the child. So again, it's always best to catch this early and to treat them. Another example of a spirochete is going to be Borrelia burgdorferi. And Borrelia burgdorferi causes Lyme disease. And Lyme disease, the way that you get Lyme disease is that it gets into your blood through a tick bite. So what ends up happening is that this tick will carry Borrelia burgdorferi. And once this tick will bite you, that will allow the organism to get into your blood. And so places where you would typically become infected with this organism would be if you live in areas that have kind of woods or forests, and if you're out and you get bit by a tick, typically the tick are found like in areas that are that have deer in them. And if you happen to get bit by this tick, that causes Borrelia burgdorferi to get into your bloodstream. And the characteristic stage or the characteristic sign of having Lyme disease is that it starts as this bullseye-shaped rash. So notice you have this kind of middle, a clearing, and it has another red area. So look at it looks like you would, if you're playing darts, it looks like a bullseye. The rash is about 15 centimeters big, typically in size, and this rash typically will be seen in about 75% of cases, meaning that most stages of Lyme disease start with this rash. What then happens is flu-like symptoms appear in a couple weeks as the rash fades. So patients might get fatigued, tired, etc. Antibiotics taken during this interval are very effective in limiting the disease. Again, meaning that if you catch it early and you treat it, it's much more likely to be resolved than if you let it keep progressing. During second phase of Lyme disease, that's when the symptoms become more incapacitating. And so the heart becomes affected, and oftentimes a pacemaker is going to be required to help an irregular heartbeat. You might have incapacitating chronic neurological symptoms such as facial paralysis, fatigue, memory loss, etc. And in the third phase, sometimes arthritis appears or swelling of the joints. And so again, catching Lyme disease early and treating it is going to help with the prognosis. And so the way that we make a negative stain, again, is going to be a little bit different than how we would make a simple stain. And so what you would do is you would take your slide and you would want to label on the frosted part what is going to be on your slide. So remember, when you do this, you're going to put frosted side up. You're going to label the slide with your name, so your initials, the date, negative stain, right? So you know what kind of stain is on this slide. And then you're going to test along your gum line. And so maybe on your slide, you would label gum line. What you're going to do is you're going to start by putting a drop of Congo red on your slide. Now, one of your questions in your question set says, what is wrong with the diagram found in your lab book? And the answer is that you don't want an entire drop of Congo red. You only want a half drop, meaning that when you're dispensing the dye, instead of letting the drop fall all the way out of the dropper, instead you're going to squeeze the bulb, let a little bit of dye out, and then touch it to the slide. And that's going to give you this half drop of Congo red, meaning it's not an entire drop. And the reason for that is that in this stain, remember, you can't use heat. So you're not going to air dry it on a slide warmer. You're not going to heat fix it. You need it to air dry. If you use an entire drop of Congo red, it's going to take a long time for that stain to dry. And so it's much, much better to use only a half drop. So you're going to start by putting a half drop of Congo red on your slide. Then you're going to take a sterile toothpick and you're going to run the toothpick along your gum line. So not on your tooth, don't scrape just your tooth. You don't need to poke your gum line, but just very gently run the toothpick along the gum line. Then you're going to take that toothpick where you swabbed your gum line, and you're going to stir it into that drop of Congo Red. Now they used a loop, but because we are using a toothpick, you're going to do this using a toothpick. Then you're going to take a second slide, and for the second slide, you're going to smear the dye all across your slide. And so what you do is you start by first pulling the slide in one direction. So notice they're pulling it towards the edge and then pushing it to the other end. And so what you want to do is essentially smear that Congo Red all across the slide. And once you're done with that slide that you used to smear the bacteria, take that slide and put it directly in the container that has the disinfectant. After you've smeared your bacteria, you're going to let it air dry. Again, no heat at this step, just let it air dry at your desk, and then you're going to view it under the microscope.
So what we're looking at here is, this would be on the left, a picture of an oral spirochete. So this would be if you did this experiment using the scraping along the gum lines, like in the instructions. And so some people would have a oral spirochete. And so if we do our negative stain, the background is going to be red because we use Congo Red. Congo Red is a negative stain. It's acidic, which means that it donates hydrogens to the solution, and therefore the oxychrome on the stain is going to be negatively charged. Because the dye is negative and the cell is negative, the dye is repelled by the cell, meaning the dye is not going to stain the cell, but instead will stain the background. And so when we did this, we look at the oral spirochetes. And so again, we can see these little spirals, and these are a type of bacteria that could be found in your mouth. Now, does that mean that you have syphilis in your mouth? No, there are other types of bacteria as well that are spirochetes. And so some people would have these and others would not. On the right, we just have a different example here. This would be if we did a negative stain of Staphylococcus epidermidis, and you can actually see the morphology here and the arrangement. So remember that Staphylococcus, coccus is round or spheres, and Staphylo refers to the fact that it's these grape-like clusters. And so you can see that the Staphylococcus epidermidis is stained colorless, meaning it's not stained, and the background is what's going to be red. And so I've posted another video that is a demonstration of somebody actually doing the negative stain. A couple differences between how they're doing their procedure and how we would do ours. One is in the video that you're going to watch, they are using nigrosin, which is just a different type of negative stain. Whereas if we were doing this in our lab, we would use Congo Red. Another difference between what our protocol said versus what you're going to see in the video is that in our example, we were looking at scrapings along the gum line like we have for our oral spirochete here. In the video, he's doing the experiment using bacteria that's on a plate. But otherwise, the technique is the same, and so it's just a way for you to visually see the way that the negative stain works. And so you'll want to watch that in addition to this to just see how the procedure is done. And so that will conclude our lab on the negative stain.